Good morning, everybody. This presentation is on an article called Nitrogen Fixing Organelle in a Marine Alga, and it is a study done on a microbe called UCYNA. This microbe was originally thought to be a nitrogen-fixing cyanobacteria in endosymbiosis, but this study shows that it has in fact evolved to be the very first documented nitrogen-fixing organelle. What is UCYNA? Candidatus, a telocyanobacterium thalassa, or UCYNA, has historically been thought to be a cyanobacteria that's in symbiosis with a haptophyte alga called Brutosphera bigelowi, or B. bigelowi. In this photo here, you can see this is a B. bigelowi, and each one of these has only one UCYNA per. So you can see it here in sort of the center where the black arrow was pointing to. There are two different ecotypes of this microorganism which allow it to occupy both coastal waters and the open ocean, and it's found across the globe, even in Arctic water. Both of these ecotypes have been shown to have the same evolutionary characteristics of an organelle. So what was tested and how was it done? Previously, this has only been studied using 2D imaging, which did not give scientists the ability to determine the exact location of UCYNA in B. bigelowi, and it was hard to determine how it divided. But this study used soft X-ray tomography, or SXT, which creates high-resolution 3D images. And this photo down here shows you what that would look like, an example of it. So you can see all of the components and where they are in the cell. This is a nucleus here. And they also used linear absorption coefficient, or LAC, which measures how much radiation is absorbed by different components in the cell to determine changes in density and composition. Hundreds of specimens were imaged using SXT, and 49 of those specimens were then segmented to measure volume and density during fission. Okay, so this image here is broken up into 3 different sections. Part A shows the 3D images of the light cycle, where you can see all the different components in the cell, the UCYNA, the nucleus, the mitochondria, and the plastids. Section B shows the 3D SXT, soft X-ray tomography renderings of the dark cycle, and shows replication and fission of UCYNA and B. bigelowi organelles. Section C is the LAC measurements of density and composition for all of the components, the mitochondria, UCYNA, nucleus, and the plastids. And so what is happening here during the dark cycle? So during the first half, there is doubling of the mitochondria, and then it splits into 2. The UCYNA replicates, and fission occurs, and it goes into 2 separate bodies with decreased density. And then the host cell's nucleus and plastids split to create 2 identical daughter cells. And cytokinesis occurs, so now we have 2 specimens, each having its own UCYNA. And then through the second half of the dark cycle, all of these components continue to grow. This study also compared the protein levels in whole symbiotic cells to isolated UCYNA. Most of the protein matched the genome that they had come from, but about 368 proteins found in UCYNA were B. bigelowi encoded, suggesting that UCYNA imports proteins from B. bigelowi. It has been shown that UCYNA is unable to make certain amino acids, nucleotides, vitamins, and cofactors on its own, and so it imports proteins from its host in order to complete these biosynthetic pathways. Because of this, it has been suggested that UCYNA's genome has shrunk due to redundancy, and importing proteins from its host is the behavior of an organelle, which is further proof that UCYNA has evolved or is evolving into an organelle. So this image here is also from the paper. It shows the metabolic pathways in UCYNA. The green outlines are UCYNA encoded. They're only found in UCYNA. The purple filled ones that have a purple outline with the purple fill, they are missing from UCYNA, and they're imported from B. bigelowi to complete the biosynthetic pathways that they're in. And then the green outlined ones with the purple fill are redundant proteins, and they are found in both UCYNA and B. bigelowi. And then on the right here, you can see the proteins that are more abundant during the day including the NIF gene, which is here, showing that UCYNA does nitrogen fixation during the day. And all of the proteins that have asterisks here show that they are involved with nitrogen fixation and metabolism as well. Scientists are calling this new organelle a nitroplast, and it is the first nitrogen-fixing organelle ever found so far. This organelle is similar to a chloroplast, but instead of fixing carbon, it fixes nitrogen. This study shows that UCYNA has been tightly integrated into the cellular structure of B. bigelowii, replicates with the host during the dark cycle, has extensive gene loss due to redundant genes between the two, and that it imports proteins from the alkyl genome. So what does this mean? Scientists have been working with directed evolution and metabolic engineering to produce nitrogen-fixing capabilities in agricultural plants. And this has allowed for optimizing nitrogenase to fix atmospheric nitrogen in plants and engineering rhizobia for symbiotic relationships with non-legume plants. This engineering could allow plants to grow in extreme conditions such as arid deserts, saline alkaline soils, and even on Mars. So everyone pack your spacesuits! Thank you very much for listening. I hope everyone enjoyed this presentation on a new organelle called a nitroplast.